Greetings, everyone. My name is Oluchukwu and my topic is preliminary in-silico drug repurposing attempts for management of Parkinson's disease. These are my outline. Introduction. We know that Parkinson's disease is one of the neurodegenerative movement disorders that affects mostly elderly people. And one of the profound effects of this Parkinson's disease is loss or slow movement. This is because of the loss of the dopaminergic neurons. And we all know that most of the drugs used in management of this disease don't really slow down the progression of this disease. Instead, they only help in managing the signs and symptoms of the disease for temporal effects. That's why we had to undergo drug repurposing, which is finding new applications for medications that are already in use. Statement of problems. The most significant unmet therapeutic need in Parkinson's disease is the development of intervention capable of slowing down this disease progression. We know that most medications in use only elevate the symptoms. That is, they help temporarily in slowing down the signs and symptoms of this disease, not really halting the disease progression. Aim of this study, the study aims to examine and screen already existing drug compounds distinct from those already established, which has anti-Parkinson effects. These are my specific objectives. Justification of the study. The development and discovery of compounds that will reduce the progression of this disease remains the priority for patients and researchers. The increase in the number of failed clinical trials caused an increase in the length of the drug development process has led to the use of already existing compounds meant for other indications as novel treatments in Parkinson's disease. This is where drug repurposing comes in, whereby we use drugs meant for other disease in treatment of a new disease. These are my materials. Method. First of all, we did literature mining to get the receptors and the target sites for this disease with selection and preparation of validation of the receptors. Another method is selection and preparation of the ligand, validation of docking protocols, docking simulation, and these are my results and discussion. And we worked with 3 proteins, which are targets, which we downloaded from Protein Data Bank, and the ligands, which are already existing drugs. We had to download these drugs from DrugBank online. And we also carried out a molecular docking simulations. And from the table stated above, these are the drugs that have higher binding energy than the reference compound listed below. The first one is for protein X8XIC. The next one is for protein XIW, and the next one is for 7JV5. And we also analyze these drugs, which are frontrunner drugs, to determine the CNS-like physiochemical properties of this drug. We carried out this analysis using Datawarrior software and worked based on the targets or criteria that has been enlisted for CNS-drug, that is drug that pass through the central nervous system. Some of the criteria that this drug, which are frontrunner drugs, needs to meet was the molecular weight, which should be less than or equal to 450, the minimal hydrophobicity, the number of hydrogen bond donor less than 3, the number of hydrogen bond acceptor less than 7, the number of rotatable bond less than 8, hydrogen bond less than 8, and polar surface area less than 60 to 70 Armstrong. And we also carried out SwissADME analysis, which is to be able to ascertain the pharmacokinetic properties and drug likeness of these drugs. And the drugs, which are frontrunner drugs, after analyzing them with Datawarrior, we were able to, from the 30 drugs that has higher binding affinities or energy, we were able to obtain 10 drugs, which are frontrunner drugs, which were analyzed using the SwissADME tool. And from it, we noticed that the pelin fails one of the lipstick rule of 5. Hence, it doesn't pass the BBB, which is the blood-brain. The boiled egg approach was also used to ascertain drugs that can pass the BBB, which is the blood-brain barrier, and drugs that can pass the gastrointestinal tract. And from our results, we noticed that adapalene is the only drug that is outside the egg yolk, which shows that it doesn't. The amino acid interaction of these frontrunner compounds we analyzed with the reference compound and drug already existing in the market, which is levodopa. From our results, we noticed that levodopa was 67%, has a 67% amino acid interaction, same with ozadrolone and hexamistine. In conclusion, oxandrolone and hexamistane has potential medication that can be used in treatment of Parkinson's disease. Further analysis should be done in vivo and in vitro to validate these results. These are my references. Thank you for listening.